Good afternoon, student. Today I am going to share with you the next type of asexual reproduction. Okay, that is of tissue culture. So, what is the tissue culture? Tissue. What is the tissue? Tissue is a small piece of cells. Clear? A small piece of same type of cells is known as a tissue. Clear? When same type of cells bind together, that time it forms tissue. And when same type of tissue binds together, it forms a organ. When different organ works together, it forms a organ system. So this is the main sequence of life as we can observe it in our surrounding. Clear? So here is a one method of plant reproduction in which we can understand that how tissue culture takes place. Okay, so we can observe that plant can grow from a small piece of plant or a single cell also. Clear? Several times there is a modification of methods by which we can produce plants from a single cell or a mass of cell, that is only known as a tissue culture. Clear? सेम सेल्स का जो ग्रुप होता है उसको टिश्यू कहते हैं. वो टिश्यू के एक पीस से हम पूरे प्लांट्स को बना सकते हैं. दैट मेथड इज नोन एज द टिश्यू culture. Okay, the production of new plants from a small piece of plant tissue, clear, removed from a parent plant, and if we provide a suitable growth medium to the piece of plant, then we can produce number of trees by this. Okay, and this method is known as the tissue culture. Clear? As you can see on your screen also, first what we have done, suppose a plant, from a plant we can take out tissue from the piece of leaf. Okay, and we can directly isolate some cells from it. There's two methods. Okay, if we grow them or dip them in a culture medium. What is this culture medium? Culture medium is having that a main component is of agar. Clear? Agar is extracted from algae. Clear? All the plant nutrients, agar, plant hormones, whatever things which is necessary for the plant growth is placed inside this petri dish. Clear? So this solution, the culture solution, contains plant nutrients also, agar also, plant hormones also, because they all are the necessary things for the growth of a plant. Clear? If we place several pieces of plant in it or some cells of plant in it, after few days only we can observe that the small saplings come out from the piece of tissue. Clear? These saplings can be transplanted in nursery, or from the nursery we can transplant them in fields. So this is the method by which we can produce several plants from a piece of plant tissue. Okay, so this is the method of plant. Again I am telling you what is the process and what are the steps of it. Okay, at first you can see the tissue sample scraped from parent plant. That is the first step. Means a small piece of plant tissue is taken from the growing point of the plant, maybe tip of the plant, maybe root tip of the plant, maybe root tip of the plant, or maybe shoot tip of the plant. Clear? Then what we do, we just place it inside the culture medium. Second step, tissue sample placed in agar's growth medium containing nutrients and auxin. Beta 1 chapter is deleted from your syllabus, but in that also auxin has introduced. Auxin is a one kind of plant hormone which is very important for the growth of plant. Clear? Oxygen, gibberellin, cytokinin, these are the abscisic acid, these are the plant hormones and have different functions in plant growth. Clear? So auxin is a one kind of plant hormone which is necessary for the growth of plant. So agar solution contain oxygen also. In third step, samples develop into tiny plantlets. Okay, so this is the mass of cell. When mass of cells grows, that is only known as the callus. Means the hormone which is present inside it, like auxin, these hormones make the cells in the plant tissue develop very rapidly. Okay, the development of plant cells or division of plant cells is very rapidly. So that time a group of cells formed here. Clear? That is the shapeless lump of mass, and this shapeless lump of mass is known as the callus. Clear? The callus is then transferred into jelly containing the callus develops root and shoot. Clear? So first this callus has to be transferred to another petri dish. Clear? The callus with developed roots in then put on a yet another jelly containing substances. Different hormones are there. The callus having root and shoot separate into tiny plantlets. The callus is then transferred into, and the inside of it the plants is having roots and shoots, and it forms a small plantlet form. Clear? Then the plantlet thus produced are transplanted into pots or soil, that which I have told you that after when the plantlets develop in a proper form, these plantlets planted into the compost or soil. Okay, so this is the method by which we can produce ornamental plants, orchids, dahlia. Clear? And other plants also. The production means ornamental plants means which plants provide us benefit. The production of plants by the method of tissue culture. This the whole method is known as the micropropagation. Sometime in exam it has asked that in place of plant tissue culture they use the term define micropropagation. So that time your answer is just similar with the tissue culture, because both the processes are similar. So what are the advantages of tissue culture? The main advance of tissue culture is very fast method. Clear? Thousands of plantlets can be produced from a small piece of plant tissue. Here only you can see that from the one plant piece we are able to grow that much plant. So just think, if we use it in a commercial way, so how many plants we can produce from a simple small piece of plant. So the new plants produced by tissue culture are disease free, because if we want to grow something, we always choose a best one plant. If we are choosing healthy plant, definitely all the plants which is developed from healthy plant, they will also disease free, or they will also be healthy plant. Okay, now tissue culture can grow plants round the year, means any season, any weather, any condition we can grow plants because of the tissue culture. Clear? Very little space is required. You can see here on picture only, you can observe that a small petri dish is more than enough for the development of it. When the small plantlets get roots and shoot, that time it can be transferred from another area. Clear? So very little space taking consuming method it is. So plant tissue culture is very important, and it is a very nowadays it's a very invoke process. Okay, means it is using day by day in a laboratory condition, a very sterilized condition we are using this method. So it's a very less chances that the new plantlets get any kind of infection. Okay, thank you children. If you have any question regarding this, you can send me your question.